Okay, let's unpack this. Imagine trying to find just one, perfect molecule.
Out of billions of
Right.
possibilities.
Billions. And hoping it becomes a life-saving drug. It's just this monumental task.
Uh-huh. It takes years, costs, well, astronomical sums.
We're talking over a decade sometimes.
Mhm.
And billions of dollars.
Easily. And even then, when you think you've got a promising candidate, most of them just fail.
Yeah, and a huge reason for that failure, a real bottleneck, is figuring out how that molecule actually behaves once it's inside the body.
Exactly. We call them its ADMET properties. It's an acronym for how well it's absorbed,
Where it distributes.
how the body metabolizes it, how it's excreted.
And, crucially, toxicity. Does it actually work without causing harm?
That's the million-dollar question. Or billion-dollar, maybe.
Right. And traditionally, figuring all this ADMET stuff out means lots of lab work. Slow, expensive lab work.
Oh, yeah. Think cell cultures, animal studies. Each one takes time.
Uses up your precious compound.
And just racks up the costs. It's a slow, laborious process.
This is why the field's been pushing so hard towards in silico methods, right? Computational approaches.
Exactly. Using AI, machine learning, to predict these ADM properties before you even make the molecule in the lab.
That sounds like a massive shortcut.
It really is. High throughput, much lower cost, and way faster. You can screen thousands, maybe millions of virtual compounds computationally.
Okay, but even with machine learning, there's still a catch, isn't there? It's not just plug and play.
Not quite. Building a good predictive model, one you can trust, isn't just about feeding it data. You have to pick the right type of algorithm.
Like random forest or support vector machines, things like that.
Precisely. And then you have to fine-tune all its settings, its hyperparameters. This manual tuning part is incredibly complex.
Right, it needs deep expertise. And you're sort of sifting through endless possibilities, like trying to guess a combination lock code.
That's a good analogy. And that complexity, that tuning headache, is exactly where automated machine learning, or AutoML, comes in. It's potentially a real game changer.
And the research paper we're looking at today dives right into that, exploring how AutoML can tackle this tuning problem.
Yeah, they focus specifically on one AutoML method called hyperopsclern, or HSL.
Okay, HSL. And their goal for this deep dive, using the data, was pretty ambitious. They wanted to use HSL to automatically build models for, 11 different ADM properties?
11 critical ones, yes. They gathered big data sets covering key areas. For absorption, they looked at CACO2 permeability. That's about crossing the gut barrier.
Okay.
And also PGP substrate identification, basically. Does the drug get pumped out of cells?
Important stuff. What about distribution?
They looked at blood-brain barrier permeability. Super important if your drug needs to get into the brain.
Makes sense. And metabolism.
That was a big focus. Predicting inhibition of the 5 major CYP enzymes. These are the enzymes that break down most drugs in the liver.
Right, you don't want your drug messing those up.
Exactly. Plus, they looked at stability in liver microsomes, both human and rat, and for toxicity.
The T in ADMET.
They included HERG inhibition, which is a standard flag for potential heart rhythm problems.
So a really comprehensive set of properties. And instead of researchers manually trying out all those different algorithms and tweaking settings for ages,
They just fed the data, essentially, into this HSL tool. The AutoML system automatically searched through all those algorithms and their settings.
it did the complex tuning part automatically.
That's the idea. And the results, honestly, pretty fascinating. These automatically generated models performed remarkably well.
How well?
Across all 11 properties, the models got an area under the ROC curve and AUC greater than 0.8.
Okay, remind us what AUC means again.
Sure. It's basically a score from 0 to 1, measuring how well the model tells the difference between compounds that have the property, say, being toxic, and those that don't. 0.5 is just random guessing.
And one is perfect.
Right. So getting over 0.8 is generally seen as a strong model. And for some properties, like predicting blood-brain barrier permeability and inhibiting one of the CYPs, CYP1A2, they actually hit AUCs over 0.9. Really quite good.
Wow. But the crucial thing is, did these models work on new data? Data they hadn't seen before?
Yes, and that's absolutely critical. They tested these HSL-generated models on external data sets, data from other labs, other studies. That's the real acid test.
So how do they hold up?
They held up very well. On these external sets, they performed comparably to, and in several cases even better than, established online prediction tools people use, and even manually tuned models from other research papers.
Better. Even better than manually tuned ones sometimes.
Yeah, for instance, their model predicting liver microsome stability was more accurate on an external test set than a published deep learning model, which you'd think would be quite sophisticated. That's a pretty significant win for the automated approach.
Okay, so let's connect the dots. The big takeaway here seems to be that AutoML, using this HyperOps learn tool anyway, it isn't just theoretical. It can actually build high-performing, reliable, predictive models for these really important ADMET properties.
Automatically. And they work well even when tested on new, unseen data. That's the key message.
And the significance of that.
Well, it's potentially huge. This kind of approach could genuinely speed up the very early stages of drug discovery. It streamlines those essential computational predictions.
Making sophisticated machine learning more accessible, maybe.
Exactly. Even for researchers who aren't, hardcore machine learning experts, they could potentially get really good models without needing to do all that complex, time-consuming manual tuning.
Right. So here's something to think about then. If we can reliably automate this really complex, expert-driven process, building accurate models for how a drug behaves, how might that change where the creative energy goes in drug design? Could it fundamentally shift how we design drugs right from that very first molecular idea?